welcome to week 4. In this week, we are going to learn about enzymes, which are nature's catalysts. So, in this first lecture, I will introduce enzymes and I will talk about how enzymes speed up chemical reactions. I will introduce the concept of active site of an enzyme. I will talk about Michaelis-Menten kinetics and Lineweaver-Burke plot, which is the linearization of the Michaelis-Menten plot. So, it turns out that proteins can be classified according to their functions. So, one type of proteins are enzymes which catalyze chemical reactions. There are 1300 different types of enzymes in human bodies. I will talk about a lot of enzymes in week 6 when I talk about metabolism. Apart from that, there are regulatory proteins which bind to receptors, for example, hormones. In the previous week, we saw examples of these FGF receptors and the factors, growth factors. So, they are regulatory proteins. In the last week, we also saw the example of hemoglobin, which is a transport protein. We all drink milk and egg and eat eggs. So, these contain casein in case of milk and ovalbumin in case of eggs. These are storage proteins. Then in our body, we have muscles which are contractile and motile proteins. So, they help us to do all sorts of work. There are structural proteins like collagen, keratins in our skin and hair, elastin which forms our vocal cord. So, these are all structural proteins. Then there are binding and interaction proteins, for example, proteins that bind to other proteins and result in signaling. So, phosphorylation of insulin receptor substrate proteins. So, we will see such signaling events a lot when I talk about cell cycle. And then finally, there are proteins which are protective proteins like immunoglobulins, which play a very important role in our immune system. So, we will see proteins of this type in week 9 lecture on our immune system. So, today I am going to focus on enzymes. Enzymes are the catalysts of nature. So, just like catalysts that we design in the lab, nature has designed these catalysts so that reactions which do not occur or which are very, very slow under normal conditions of our body temperature and neutral pH, which do not occur, they can actually occur within few seconds because of these enzymes. So, it turns out that most enzymes are proteins with exception of few catalytic RNA. We have already seen one example, which is the ribosome, where ribosome is the one which catalyzes the formation of the peptide bond in proteins, and there the active site has RNA. So, it is the ribosomal RNA which catalyzes that reaction. So, what are catalysts? They alter the rate of a chemical reaction without themselves undergoing any permanent change in structure. So, the catalyst or the enzyme is regenerated at the end of the chemical reaction, which means that they can take part in multiple reactions one after another, right. In case of enzymes, the catalytic activity depends on the folded state of the protein, and if the folded state is lost, then its catalytic activity is also lost. So, just like proteins that we have seen in the last week, enzymes have all the hierarchy of protein structure, that is, they go from primary to tertiary, and in some cases they will also have quaternary structures. Enzymes, they are found in various sizes. They can range from 12,000 Daltons to over 1 million Dalton in size. So, these are some of the reactions that are very important reactions that occur in our body, and their half-life is listed here. So, what is half-life? It is the time that is needed to reach 50% of the reaction, and that is denoted as T half. So, you can see there are some reactions where the half-life is 1 year, and then there are reactions where it can go up to 1 billion years. So, let us take the example of peptide hydrolysis. So, a peptide, if it hydrolyzes on its own, it takes around 400 years, right. The half-life is around 400 years. Now, for the reaction to reach almost 99% completion, you need around 5 half-lives, right. So, if it is 400 years or 450 years, then 5 times of that. So, 400 times 5 will be 2000 years, right. So, all these reactions are color coded according to the bonds that are cleaved. So, you can see that the red ones are the carbon-carbon bonds, and these are the ones which are most difficult to cleave, and we will see some examples in subsequent lectures. So, let us take up these macromolecules. Peptide hydrolysis at room temperature, half-life is 400 years. If we increase the temperature, of course, the reaction will become faster, but still it will be 5.5 weeks. Sugar molecules, polysaccharides, they are very stable. So, 4.7 million years, that is the half-life. RNA, we know that among all these macromolecules, RNA is the least stable. So, its half-life is also less, around 4 years, and DNA hydrolysis takes place in 1400,000 years, right. So, what happens when there is enzyme? So, these are some of the enzymes that are listed here. So, let us take the example of peptide, DNA and sugar molecules. So, you can see that their half-lives are very high. So, the reactions are very slow, but when they are catalyzed by enzymes, so these are the enzyme catalyzed reaction rates, all of them become very fast and their half-life is in seconds. So, for example, for protein, the half-life is around 500 seconds, right. So, so the rate constant is 500 per second. So, the half-life is 2 milliseconds, which means that the reaction will reach 99% completion in 10 milliseconds, which is very, very fast. So, enzymes have this impressive ability to speed up reactions which are very slow under normal conditions to such impressive rates, so that they can be completed within seconds. Not only that, enzymes also have the ability to catalyze some really complex reactions. So, we will see one example here. The enzyme oxidosqualene lanosterol cyclase. So, this enzyme results in the production of a sterol which is called lanosterol, right. So, this is an intermediate in cholesterol biosynthesis. Lanosterol is produced from this linear molecule. So, you can see that this is actually a linear molecule which is drawn in this folded manner. So, it is a 24 carbon long chain, and from that this molecule is formed which has 4 cycles, right, and it undergoes this series of reactions. So, in the first step, the first ring is formed, then the B ring, then the C ring, then the D ring. These 4 rings are formed consecutively to give rise to this final molecule, and all of these steps are carried out by this single enzyme, right. Not only that, this final product, you can see, if you count carefully, you will see that there are 1, 2, 3, 4, 5, 6, 7 and 8 chiral centers. So, the enzyme not only catalyzes this series of complex reactions, but it also forms the correct isomer at the end of the reaction with almost 100% accuracy. So, that is the fantastic ability of enzymes, that they can speed up reactions and they can also perform really complex reactions under very normal conditions with very high accuracy. So, this is the structure of the enzyme that I just discussed in the previous slide, and it is a membrane bound structure. So, we will talk about cell membrane in details. So, normally you have this polar head group and the hydrophobic chain. So, that is one leaflet of the membrane, and that is where this membrane embedded part of the protein is there, and the active site is somewhere here, right. And hopefully you can recognize by now that these spiral structures are the alpha helices and these arrow like structures are the beta strands. So, these are the secondary structures of this protein, and this structure was solved by X-ray crystallography in 2004. So, how do enzymes carry out these reactions? How do they speed up reaction, and how do they carry out such complex reactions resulting in such stereoisomers with high accuracy? The reason for that is that enzymes are big molecules, but they have a particular region where the reaction or the chemical reaction is carried out, and that is called the active site. In this case, the reactants are called substrates. So, substrate will bind the active site in a very precise manner. The reaction is taken place in this bound state where the reactant gets converted to the product, and once the products are formed, they diffuse out. So, how do these substrates bind the active site? They bind the active site because there is a huge complementarity between the substrate and the active site. So, you can see that there is a shape complementarity. So, whatever this shape is, this looks complementary to that, so that it can fit nicely to the active site. Once it sits there, there are also interactions which are complementary to each other. For example, this H denotes a hydrophobic side chain. So, it can be a methyl group, it can be an aromatic side chain which is present in the substrate, and similar side chain is present in the enzyme. So, hydrophobic interactions take place. There is a negative charge here and there is a positive charge here on the enzyme. So, when the substrate binds, we get this charge interaction, which also makes it favorable. More hydrophobic interactions here, and then also hydrogen bonding interaction. So, this is a hydrogen bond acceptor, there is a hydrogen bond donor here. Similarly, there is a hydrogen bond donor here, there is a hydrogen bond acceptor on the enzyme. So, together they will allow this substrate to bind this enzyme in a particular orientation, which helps in the specificity of the reaction, and also they will help in speeding up the reaction. Substrate binding, as we have seen in the previous week, can be of two types, lock and key and induced fit. So, as we saw in the previous lecture, lock and key can be is similar to a lock and key reaction, where the substrate has perfect complementary structure to that of the active site, right. But in case of induced fit, the enzyme structure is flexible, and once the enzyme and substrate forms a complex, there is readjustment resulting in slightly changed structure. And this shape change can help in the catalysis by stabilizing the transition state. So, I will discuss about this in more details in the next lecture. So, here is the pictorial diagram of lock and key model and the induced fit model. So, you can see that the substrate has a perfectly complementary structure to the active site, so that it fits nicely, whereas in the induced fit model, this and this are slightly different. So, once the complex is formed, it induces this structural change. So, both the enzyme and the substrate have changed their structure, and then the reaction happens and we get the enzyme plus product. So, the enzyme is regenerated at the end and we get the products. So, what are the factors that influences reaction? Just like any chemical reaction, temperature is one factor that will influence enzyme activity. So, we know that as temperature is increased, the reaction rate is increased. So, for most enzymes, if we think about enzymes that are present in humans, we will see something like this, that with increase in temperature, the activity increases. However, after a certain point, the activity decreases, and this is because when you increase the temperature beyond a certain point, the enzyme becomes unstable and it can start denaturing or unfolding, and that means that the active site will lose its characteristic, and of course, it will not be able to catalyze the reaction. So, of course, there will be an optimal temperature. So, for most enzymes in our body, the optimal temperature is around 40 degrees centigrade, because our body temperature is 37 degrees centigrade, right. But if we take bacterial enzymes, let us say thermostable bacterial enzymes, in that case we will see that their optimal temperature will be shifted to much higher temperature, like 70 or 80 degrees centigrade, because those bacteria, they survive at much higher temperature, and hence the enzymes in those bacteria have evolved to have optimal temperature at a much higher temperature that matches their environment. Similarly, pH also influences enzymatic activity, and we have already seen that all these different side chains have these different pKa values. So, depending on the pH at which the enzyme has to work, these different side chains will be found in the active site. So, some examples are shown here. For example, alkaline phosphatase or trypsin, they work very close to neutral pH, because they are found in environments where the pH is neutral, around 7.4. But pepsin, it is found in our stomach where the pH is very low, it is less than 2. Now, pepsin is optimized such that its optimal activity matches that pH, and this is something that is found quite often in different enzymes. But one may wonder that these are the side chains, so these are the functional groups that are present, but then enzymes, they tend to catalyze so many different reactions. So, how do they catalyze such reactions using such a limited number of functional groups? So, it turns out that enzymes recruit other molecules to carry out specialized reactions, and these molecules are called cofactors. So, enzymes bind cofactors at their active site, and cofactors can be classified into two, organic and inorganic. So, most inorganic cofactors are these minerals which are found in active sites, and we have already seen example of magnesium in case of our polymerases, right. So apart from minerals, there are also organic molecules which are called coenzymes. They can be either covalently or non-covalently linked to the enzyme active site, and most of these coenzymes are derived from vitamins. So vitamins that we take as a supplement from our different food, they undergo some changes and then they are incorporated into enzymes for carrying out their function. Two examples I am citing here. One is vitamin B3, which is a precursor for NAD plus or NADP plus, which are coenzymes used in different oxidation reduction reactions. Similarly, vitamin B2 or riboflavin is the precursor of FAD, flavin adenine dinucleotide, which is also important in many enzymes that catalyze oxidation reduction reactions. I will talk about vitamins and these coenzymes in more details when I talk about metabolism in week 6. Okay, so let's look at enzyme kinetics. Based on the diagram that we saw in one of the previous lectures, previous slides in this lecture, so we have the enzyme plus substrate which forms the enzyme substrate complex. Once it's in this bound state, we have this enzyme product complex. So the reaction happens here. This is where the real chemistry happens, and then the product dissociates. We can consider all of these as reversible steps. So we will have rate constants for all of this, which turns out that we will have 6 rate constants. But we can actually simplify this to something like this, because this will be a very fast step and this reaction step is also quite fast. So we can combine these two steps into this one irreversible step where the enzyme substrate dissociates into enzyme plus product. So this type of approximation was done by Leonor Michaelis and Maud Menten more than 100 years back in 1912. So that is when their paper came out, and they showed that enzyme kinetics can be explained very nicely by a very simplified kinetic reaction like this. So K1 and K minus 1 are the on and off rates of the substrate binding, and K2 is the rate in which the enzyme substrate complex is converted to the product and dissociation. So if we have this equation, we want to know what is the rate of product formation. So rate of product formation is also called the velocity, that will be given as dp dt, or the rate of product formation. So if we look at this equation, the rate of product formation will be this rate constant multiplied by the concentration of the enzyme substrate complex. So K2 times ES. But of course, we do not know what is ES, what is the concentration of the enzyme substrate complex, and that is what we want to figure out. So if we look at the rate of formation of the enzyme substrate complex, which is ds dt, from this equation we can write it as, so the rate of formation is this reaction, so K1 times free enzyme plus free substrate, which is this, that is how it is formed, but then it is destroyed by this and this, these two steps. So K minus 1 times ES, which is this, and K2 times ES, which is this. Now, since these two steps decrease the concentration of ES, we have negative signs here. So this is something that was not known when Menten and Michaelis-Menten, they came up with this equation, but now using numerical analysis, we can actually plot the concentration of the substrate, the product, and the enzyme substrate complex. So of course, the substrate starts with some value at time 0 and it will decrease over time. The product will start from 0 and it will increase over time. But this enzyme substrate complex shows a very interesting behavior. It will start with 0, it will quickly increase to a particular value, and then it remains steady or constant at that value and then finally decreases. So this period of time is where this enzyme substrate concentration does not change. And Michaelis-Menten, they correctly assumed that there will be a steady state where the enzyme substrate concentration will not change with time, as a function of time. So dEs dt will be 0. So let's say this is our equation 2. So this dEs dt is 0 at under this steady state condition. What we want to do is we want to find the velocity which is dp dt, and that equals to k2 times Es. So, what we want to do is we want to solve for Es from this equation. But here you see that we have Es and then these constants are fine. But here we have free enzyme concentration and free substrate concentration. So, we do not know the concentration of free enzyme. We can replace that by this, because we know the concentration of total enzyme, that is something that we have added in the experiment, and that will be the summation of the free plus bound enzyme. So, Es or the free enzyme E will be equals to E total minus Es, and we can substitute that here. So, that is what is done here, and then if we separate all the variables, we will get the enzyme substrate concentration as E total times the substrate concentration divided by Km plus substrate concentration. Now, what is this Km? Km is K minus 1 plus K2 by K1. So, when we do this separation of variables, we get that. Now, since all three are constant, so instead of writing it like this, we can combine them together into one constant, which we denote as Km, where M stands for Michaelis and Menten. So, again coming back to this V, which is dp dt equals to K2 times Es. We know what is Es, so we substitute for that. Now, if you think about this, the velocity equals to K2 times Es. K2 is a constant and Es is the enzyme substrate concentration. This will have a maximum value of E total, because the maximum that can happen is all substrate are bound to all the enzyme are bound to substrate. So, Es can have a maximum value of E total. So, K2 times E total can be denoted as the maximum velocity. So, that will be the maximum rate of product formation. So, we can replace this K2 times E total with Vmax, and this is the final form of the Michaelis-Menten equation. So, if we plot this V naught versus the substrate concentration, it looks something like this. And how do we get this plot? We get this plot by doing a series of experiments. So, for a particular substrate concentration, we will calculate the rate of formation of the product, right? Or so, this is the rate of formation of the product. You can see it starts from 0 and it will saturate. Since we want the initial velocity, we will take the initial slope here. As we increase the substrate concentration, this slope will increase, which means that the dp dt is increasing or the velocity is increasing. Remember, in all this series of experiments, we have to keep the enzyme concentration exactly the same. We are only increasing the substrate concentration. So, from this series of experiments, different velocities are obtained for different substrate concentrations, and those are plotted here. So, velocity will be plotted here and substrate will be plotted here, and it gives us a curve like this. So, if we look at this curve, when the substrate concentration is very high, it will be much more than Km. So, V naught will be equals to Vmax, and that is what we get here. When the substrate concentration is low, then we get this, because S is less than Km. So, V will be proportional to the substrate concentration. So, it will increase linearly. Also, if we go to the Michaelis-Menten equation and we plot Km equals to S, we get that the substrate concentration and Km will be equal when the velocity equals to half Vmax. So, once you get this plot, we have to make sure that we reach this saturation state. From there, we will get Vmax, and then if we go to half Vmax, we can extrapolate to this and we can get the Km. So, we can get Vmax and Km, which are two very important parameters of an enzyme. But you can see that this is problematic, because if we stop here, then we will consider this as Vmax. So, our Vmax will be underestimated and our Km will also be underestimated. So, to get rid of this problem, Lineweaver and Burke proposed that we can invert this equation like so and make it a linear equation like so. So, V naught becomes 1 by V naught and S becomes 1 by S. So, you can see that 1 by V naught equals to some constant times 1 by S plus some constant. So, this is very similar to y equals to mx plus c, which is a straight line equation. So, if we plot 1 by V naught versus 1 by S, instead of V naught versus S, we will get a straight line plot, and the straight line will start from somewhere here and go up to here, right. But since it is straight line, we can extrapolate it and get the y intercept and the x intercept, and if we get these two, we can get the Vmax from the y intercept and the Km from the x intercept. So, that is the power of this Lineweaver Burke plot, and I will come back to this in the third lecture this week when I talk about inhibitors, and we will see that this plot becomes very useful in determining which type of inhibitor we have. So, Km, Kcat, Vmax, these are very important parameters. Km gives us how strongly an enzyme binds its substrate. So, this is very similar to Kd. So, you can actually compare Km with Kd and see that they both have the unit of concentration, and Km will be slightly different from Kd because it also has the constant K2 in it. Kcat gives us how fast catalysis is done, and Vmax is the rate of catalysis for a given enzyme concentration. Also, Kcat over Km is a parameter that is often used to indicate specificity of an enzyme. So, that is somewhat shown here. This is very similar to the plot that I showed before. So, these are the uncatalyzed reaction, and the Kcat over Km is plotted here. So, Kcat over Km will have the unit of per molar per second. So, this is second order rate constant, and you see that these values are all in the range of 10 to the power 6 to 10 to the power 8 per molar per second. This number is important because this is reminiscent of the rate of diffusion of molecules under normal in water at room temperature, right. So, it means that enzymes, all these enzymes, they speed up this reaction so much that the rate of catalysis is limited by the diffusion. So, how fast the enzyme and substrate diffuse towards each other and bind to each other. So, that rate determines the rate of the reaction, because you cannot speed up the reaction more than that. Once only they bind, then only you can do the catalysis, right. So, evolution has optimized these enzymes perfectly so that their rates are now limited only by the diffusion constant. So, these are called diffusion limited. So, I think that is all for today. Again, these are the books that you can refer to for this lecture. Thank you.